Okay. As Alex already talked about the slide, I'll just do a quick recap regarding this. We have a part of physiology where there is like a multi-layer mixing happening. And when we want to measure the muscles, we apply some kind of methodology. And usually we are faced with some limitations of that methodology. So what we want to do is we want to understand the physiology, but we are limited by the methodology that we selected. As we all know, the muscles are comprised of motor units that have different sizes and are kind of randomly distributed in the muscle tissue. We can see a simple scheme of a muscle with a large amount of motor units here. Of course, not all of them are active, but those who are, they contribute differently to the final result or to the final signal that we are recording. They are contributing their energy depending on where they are, how big they are, how close to the electrode they are, and so on. We can see that the motor units that are bigger or more superficial contribute a lot more to the force on the right side here. And this means that the smaller motor units that are as well present or as well active are not as loud as the bigger ones, and we are having trouble hearing them. And this means that we are kind of biased to those that are bigger, okay, or closer to the surface, closer to the uptake electrode, and so on. But before, of course, we get to the motor units, we have to. So before we talk about motor units, we have to get to them. And this is one example of what we can do. We can use a CKC method that works in a style that it iteratively estimates the motor unit filters. And when we apply the motor unit filter to the EMG signal, it yields out pulse train or a spike train of that specific motor unit. The next step here is we have to do a segmentation of the pulse trains. What this means is for each and every pulse in the pulse train, we have to decide if that pulse actually belongs to the motor unit that we are looking at, or it's just maybe basically noise. And this last step can be very time-consuming because it requires some manual inspection to do. Okay, Alex showed this slide before, but he did not explain it. What this slide actually shows is if we take a monopolar measurement, which is in the upper right corner, and apply a Laplacian filter to it, we can get a bit more sparseness in the signal. So there is some sort of sparseness, some sort of sparseness can be seen, but we still have a contribution of more motor units in the same signal. So what we can do is we can go a step further and add more spatial dimensions. In this way, we add more channels. We can see that here in the lower left part. We can see that we added a bit more channels in a spatial dimension, and we get the motor unit filter 1. And on the upper right side, we see that the sparseness increases. The next step is let's add even more channels to that. We get motor unit number 2. When we apply it, the sparseness increases even more. And when we run out of spatial dimensions, we can go into the temporal dimension. And what we do is we just add a few delayed repetitions of the signals, and this increases the sparseness even more, or even to the level where we can see the pulse trains or the spike trains very clearly. And this is what the motor unit number 3 shows. Now the question here is, what is the optimal combination of spatial versus temporal dimensions? Okay. Of course, when we get the filters, we have to somehow estimate them, if they are how well they are working, and we can use metrics like pulse-to-noise ratio, which will tell us the ratio between the energy of pulses against the noise, the baseline noise. But the trick here is that we are looking at the metrics called pulse-to-noise ratio. And here, the question always arises what the pulses are. Okay, if we would look at the pulse train where 2 motor units are merged together, the pulse-to-noise ratio will tell us that how well those 2 motor units are separated from others. So always, when reading pulse-to-noise ratio value or the assessing with pulse-to-noise ratio, always be careful to know what the pulses are. Okay. Here we can see that if we, on the left side, on the left graph, we can see that if we take a pulse-to-noise ratio of 30 decibels, the sensitivity will be around 90%. But don't take this for granted, because, as I said, we have to know what we are looking at, what the pulses are. Okay. I already mentioned that motor unit merging can be somehow present in our results. And so after we do the decomposition with the algorithm I mentioned, we get the pulse trains. And when we look at them, we can see something like this. In this slide, the merging can be maybe a bit difficult to detect, but it is there. It is marked with the red box. The question here is, could we resolve this kind of merging? Anyone experiencing that? Okay. I would say that we would have like a tough job doing it. Okay. But the next example is maybe more clear. We can see the merging as well, but it's a lot cleaner example. Here, maybe the problem would not be so tough and as it would be on the previous slide. But why does this happen? So the main reason why motor unit merging happen is because the motor units share their motor unit action potential shapes. So they have a similar, sorry, not share, but they have similar motor unit action potential shapes. They share approximately the same location inside the muscle tissue. They have approximately the same motor unit territory, share the position of veneration zone, and so on. So what we see here is for muscles with a lot of motor units, and the yellow ones are circled with identified as merged motor units. So what can we do about this? We could go and resolve the merging. Okay. If we would not be successful, we could just discard the motor units that are merged. But what would happen if we take a look at these results on the right side? If we would just discard the merged motor units, we would actually lose 2/3 of identified motor units. In this case, of course. But for the further analysis, we'll see that the number of motor units is very important. So the more we have, the better. Okay, and the question here arises, can we use them? So do we have to discard them? Do we have to identify them individually, or can we just use the merged motor units as well? And the answer here is that yes, but we have to be careful how we interpret this later on. We will talk about this a few slides later. Before we get there, we'll just discuss two mathematical operations. The first one is coherence. What the coherence means is how two time series match at specific frequency content. And the cumulative spike train is nothing else but summing together the pulse trains of individual motor units to a common or cumulative spike train. And now if we return to the previous slide, why would we bother identifying each motor unit if then in cumulative spike train we sum everything together? Okay, and that's the question. Of course, we would identify individual motor units for a specific reason, but for this reason, we would not need to do it. It is known that if we want to calculate the coherence between, I don't know, two motor pools, it is wise to do it through cumulative spike train. And this is one of the example. What happens when we sum together all the spike trains? What this does is it filters out the increasing jittering of motor units, and it yields some common component or component that is common to all motor units. Now, if we try to calculate coherence between two, let's say, sister muscles, it's gastrocnemi medialis and lateralis. In the first one, there were 43 identified motor units, and in the second one, 35 identified. And if we calculate the coherence between those two pools of motor units, with the increasing number of motor units that we include into the calculation, we can see that when we add the number of motor units, we can see it on the right side, the frequency peaks appear. Okay, so we kind of extract the hidden frequency content of motor units. We can go even a step higher and ask ourselves, should we even bother identifying motor units, or can we do something else? And yes, we can introduce the activity index into the story. And what the activity index actually is, it is calculation of CST over all of the detected motor units in the detection volume of the electrode. Okay, on these four images, there's depicted what the activity index would take into the account, approximately what, of course. Advantage of this is that we take into account all the motor units that we have taken or detected with the uptake electrode. But the disadvantage here is that we do not identify individual motor units, and we are actually not able to functionally cluster them. Okay. So the next chapter here that is like a natural next step is clustering. How can we cluster the motor units that are like firing together, merged together? We talked till now, we talked about metrics like coherence, like CST, activity index, and so on. But we can do it in a temporal dimension as well and use correlation. Okay, with correlation, we can, I don't know, calculate synchronization and so on. What we did here is we identified motor units of a trial of a recording from plantar flexion. the motor units from muscles were identified, and then instead of plotting firing rates of individual motor units, you know what I mean with firing rates. So those dashes, vertical dashes, Darius showed them before. Instead of doing that, we plot them with a line and low pass filter. And what we get is a smoothed discharge rate of that motor unit. And this is a one line that represents one motor unit with its smoothed discharge rate. And then we can use those lines, under quotes, to calculate some correlations. The question here is, I said that these signals are from plantar flexion. Okay, we know what the plantar flexion is so pushing down. Okay, and we naturally would see three muscles being active: gastromedialis, lateralis, and soleus. But we also have a tibialis active here. Why this is happening is because the persons doing or the participants executing the plantar flexion were asked to stabilize it. Okay, and because of the stabilization, when we are doing the plantar flexion stabilization, the antagonist muscles activate as well, and that's why we see tibialis. Okay, this is important because we have in the next analysis we will see how tibialis is connected to other. Okay, now when analyzing these results or these signals, we can do it in two ways. So the first one is let's calculate the cumulative spike train for each and every muscle and low pass filter it, and that's why we get 4 lines. If we go from the top to the bottom, that's why we get 4 lines in the first upper graph. Each line represents one of the muscles, and then what we do is calculate the correlations between them, and we get the simple 4 times 4 matrix on the right side. What we can read from that matrix is that gastrocnemius medialis and lateralis are highly correlated. Okay, you can see that it's the value around 1, and the tibialis and soleus are kind of correlated, and this is because of the stabilization task the person was given. But we can also see that, I don't know, tibialis and gastromedialis have something in common. Okay, we could say that they behave similarly, and soleus and gastrolateralis may be the same. Okay. And this is some info that we should memorize a bit, because we will return to that later on. We can see that these are the 3 trials for the same person, and we can see the same happening, the same results over 3 different trials. The matrix are kind of the same regarding the values. These signals are kind of special. They are from bed rest study, and these are from before 10-day bed rest study. And if we continue, these are after 10 days bed rest study. Okay. And what we can observe is that even after the bed rest study, the matrix are kind-ish the same. Okay. Of course, the values are not perfectly the same as before, but the general picture of the values is the same. Okay, so what we said until now is that tibialis and gastro have something in common. They behave similarly. And from the second analysis, we said that there's no difference between before and after the bed rest. And now, if we change the methodology, and instead of calculating the correlation between the CST of muscles, we calculate the correlation between discharge rates of individual motor units. And what we get is this big matrix on the right side, on two matrices on the right side. These red lines here depict the borders between muscles, okay? So it's the same as this on the left side. The right side is just not so evenly separated because of the number of motor units, but it's the same. So this is tibialis, gastrolaterales, medialis, and so on, okay? The same stuff. And what we can observe here is that if we take a look at the tibialis against gastromedialis, that there is no similar behavior between the muscles, but the motor units behave similarly, okay? And the next thing is that if we do a same, if we apply the same methodology on the signals from after the bed rest, we can see that, for example, if we take a look at the, I don't know, between the gastrolateralis and soleus, we can see that some new motor unit clusters are forming up. Despite the correlation from the correlation matrix from the methodology used before says that there is no difference before and after the bed rest, there is, okay? There is if we just change the methodology and calculate the correlation between individual motor units. Of course, the results have to be analyzed person-specific. We can see kind of the same pattern happening over multiple participants, but we cannot just average the results over the whole population. Okay, until now we talked about the. Just let me check it on time. Okay, we talked about the analyzing the signals that were recorded during the isometric contractions. We'll take a look at two different approaches that we also can do. The first one is dynamic conditions. So what happens in dynamic conditions is that when muscles move, geometry changes, and this complicates the analysis or decomposition in its own way. If we take a look at the simple plot of changes in the motor unit action potential shapes during the forearm extension and flexion, we can see how the motor unit action potential shape changes. So in the upper part, we can see if I remember correctly, the red line depicts fully extended forearm, and the blue line depicts the motor unit action potentials where the forearm is fully flexed. Okay, so during this movement, the motor unit action potential shape change, and what would happen is that the filter that was, I don't know, trained on the or estimated on the EMG signals from the arm being fully flexed would not work anymore because of this change. Okay. We don't need the dynamic conditions to have this kind of a problem. The same can happen in during the fatiguing. Okay, the fatigue induces the same problem. Here we have a fatigue. It's a heavy fatiguing, and we can observe the same thing happening. We have a bit of a zoom in this in one of the motor unit channel here, but you can see that the same thing happens. So how can we tackle this problem with the changing motor unit action potential shapes? What can we do about this? Can we somehow predict these changes? Can we somehow incorporate these changes into the motor unit filters? And what we come up with is tracking of the motor unit, of changing or adapting the motor unit to a specific to the change of the MUAP shapes using Kalman filter. And for example, if we would not do it, if we would like fix the motor unit filter at the shortening level 6, what the shortening level here is, it's nothing else but the movement of the arm for fully extended, fully flexed is just divided into 36 levels. Okay, so it's just this, nothing else. And if we would fix the motor unit filter at the shortening level 6 and try to use the same motor unit filter through the shortening level, we could see that at the shortening level 18, we would totally lost the motor unit. Okay, and we would say that the motor unit is not active anymore, but this is not true. It is active. It's just our filter is the problem, and the problem is because motor unit action potential shape changed so much. Okay. Oops. Okay. And the next part, and I think that this is the last one. What compensations in elicited contractions? So in the voluntary contractions, we see that the firing rates are asynchronous and more sparse. But what happens in elicited contractions, those patterns are no longer synchronous or asynchronous, sorry, and they can become very synchronized, and this possesses a problem of its own. So if we take a look at some of the example we have here, we see that in asynchronous part, on the voluntary part, we have a lot of sparseness. We have a lot of asynchronous activity. But when we go to the elicited contractions, we see that the motor unit firing times become very synchronous. And the result of this is that motor unit action potentials of motor units sum up into the compound muscle action potentials, or CMPs. How can we tackle this problem? We can estimate the motor unit filters in the voluntary part and then apply them at the illicit. Okay. We did the study a few years ago regarding this. Usually, when doing the study with elicited contractions, we study M waves H reflexes. Okay, and this is the case in the study as well. We then decompose the signals, and what we were trying to achieve here is, or to see here is, decompose the signals into motor units, then sum up the motor unit action potentials of those motor units. We get the sum of them, which is depicted by the black line, and we can see the difference between the decomposed part versus the original H reflex, which is depicted by grayish color, and we can see how much energy we have taken into account when doing the decomposition. And the same can be seen here. So we have, with the black, depicted the reconstructed or summed up potential, and with the gray color, recorded original H reflex. Okay. For conclusion, the main part here, the most important one, number of motor units matter a lot. Functional clustering matters. Okay. We have to be careful when using the activity index, because it represents the cumulative activity, and it does not support the functional clustering. Okay. And this is, I think, everything from my side.